My name is Yvonne. I work at Boston Analytical as a principal scientist. I oversee a lot of the microbiome specific projects that come to Boston Analytical. So what is Boston Analytical? We're a very strong science-based basically quality partner company. We help provide a lot of the downstream testing and characterization that drug manufacturers need to release products and to basically go into clinical trials or to sell products on the market. Basically, we're basically the people who help to ensure that the products and the drugs that are released are safe and efficacious. And a few years ago, the company invested quite heavily in microbiome-based therapeutics, specifically because this field is growing quite rapidly. This is probably the extent of the industry stuff I'll bring to this talk, just because I thought I'd go in a slightly different direction this time. So for today's talk, I will be focusing on the development of the gut microbiome and the long-term health outcomes, just drawing on a lot of the great research that people are doing. Just as Scott just mentioned, a lot of the people in the audience, you're clinicians or people who are very interested in healthcare, work in healthcare. There's literally thousands of papers available on this subject. So I'm hoping to provide kind of a small summary or a small discussion on some of the salient literature related to the development of the gut microbiome and how that impacts long-term health outcomes across the board. And then I'm basically going to start off talking about a brief recap of microbiome function. So this is going to be a little bit basic for some people in the audience, while for others it might provide some new detail. I will talk a little bit about determinants of microbiome structure. Dr. Jackson has started us off really well talking about, diet, for example. So I will continue that conversation, and then I will start talking about the development of the microbiome in utero all the way to adulthood. Again, this is going to be a very, very big picture conversation. So definitely not a full class or lecture, and I'm going to try to keep it simple and easy to understand. And then I will jump briefly onto microbiome and long-term health outcomes with a very short conversation around how microbiome can contribute to personalized health outcomes and reduction of healthcare disparities, because it's a subject that personally I'm very passionate about, and I've had mentors who are very passionate about this as well, had really great engaging conversations around. So I thought this might be a great place. George Washington is a very diverse university, so I thought it'd be a really great place to start having this conversation in the field. So let's jump in. There's a huge amount of interest in the gut microbiome, right? Scott already shared, and the opening remarks showed you there's literally thousands of publications around this. And when you move away from the literature, scientific literature, and even look at general publications, magazines, we're seeing people, even the magazines like The Economist, talking about the importance of the gut microbiome. So there's a huge emphasis on it, but why? The big question is really why the sudden, not really sudden, but it feels sudden for someone that's been doing this for over 10 years. It feels sudden that there's so much interest from the public on this. And so really, I think the reason for this is because there's now increased awareness and easily available information on how important the gut microbiome is. So this is just a recap from Scott's talk. We know the gut microbiome is the resident community of microbes in the gut. So there's fungi, there's bacteria, there's viruses, there's protists. And then we also know that the gut microbiome is an essential part of our bodies. It plays a really vital role in the health and maintenance of homeostasis. So this is a really great summary that shows you all of the different parts of our health that are touched by the microbiome, right? Even on things like you might think about modulation of bone mass density, like how does the microbiome contribute to that? But when you look at the actual way that bone density is determined, and you look at the pathway, the involvement of the immune system, and we know that the microbiome plays a huge role in the setting up, the formation of the immune system, training it, and then actually even the development of it and its full healthy function, then we can understand why the microbiome plays such an essential role in all of these things. So the microbiome protects against epithelial injury. It helps in the promotion of fat storage. It helps in the promotion of angiogenesis, And obviously GI motility and sensation, which I spent a lot of my PhD studying. The microbiome plays a very essential role here. It helps develop and train the immune system, just as I mentioned earlier. And the microbiome is one of our most important partners in the biosynthesis of vitamins and amino acids. Without the microbiome, vitamins like vitamin K would not be made, right? And then drug metabolism, we're learning more and more that there are certain drugs that require the presence of a certain profile of microbiome to work, right? And just as Scott mentioned, please bear in mind, it's not necessarily a good or a bad microbiome. It's just we're learning that there's certain types of microbiomes that work a certain way, right? And we'll get into that a little bit more later. And then the development of modification of the nervous system is has been one of the largest areas I've spent most of my career studying, how the microbiome contributes to this. Without microbiome signals, we actually wouldn't have a healthy nervous system developed across both the brain and the gut itself. And I'll talk more a little bit about this later on. And then helping to break down food compounds. This is one of the most important functions of the gut microbiome. And we've learned a lot of this from both human work, but largely using rodent models. And it's been super helpful to have animal models for basic scientists to be able to study this. So here are some of the key bacteria that and their functions, right? I just wanted to put the slide up because I thought it was a great summary of some of the really essential compounds we need in our bodies for healthy physiology and function that a gut microbiome produces. And so we can talk about something like bile salt hydrolase, which is actually produced by bifidobacteria and Bacteroides species. And so you can start thinking about if an individual has a profile in a gut microbiome that is lacking in certain species of Bacteroides or bifido that are required to produce this BSH and break down bile salts pretty much, then you start getting into, right? You can see the downstream health impacts on that individual. And even along things like lipid and glucose metabolism, secondary bile acids play a huge important role in that. And so again, using that example earlier where an individual could have, missing bifido species that makes BSH, you suddenly have more secondary bile acids, which can actually affect the person's lipid and glucose metabolism. And a lot of these pathways are involved in, general important metabolic function. So immune homeostasis and inflammation, energy harvest and fat storage, satiety control, making sure that we're not overeating. So it's extremely important, right? The gut microbiome is important. So this is why we're paying so much attention to it. So going back to the basic science part of this, these are some of the most important metabolites that we found. Doing a really quick review of literature, these are some of the most important metabolites that a microbiome makes that contribute to health. So we have folate, which, can be surprising to a lot of people to find out that microbiome makes folate. We have indoles, we have secondary bile acids, we have TMAO, which plays a huge role, not just in metabolic function, but actually when you go into neuroscience, TMAO is a very, very well characterized, and we study this quite a bit in healthy nervous system function. Short-chain fatty acids receive a lot of attention. So butyrate, propionate, acetates, these are made primarily by microbiota. So if you don't have a healthy, well, I shouldn't say healthy, a certain profile of microbiome can basically determine the amount of each of these that you make, which in the end also determine, for example, how your gut functions, because we know that colonic cells, for example, use butyrate and acetate as fuel to function. So if we have less of these short-chain fatty acids, you can begin to see, again, it's a cascade, right? So we certainly recognize that we need a certain type of profile of microbiome in order to produce these short-chain fatty acids, which are extremely crucial to health. And apart from that, we can actually then capitalize on knowing what these short-chain fatty acids do and learn how to make more of them in our bodies. And then as a neuroscientist, I geeked out over this. When I found out that, we actually have bugs or bacteria in our guts that make neurotransmitters. And so GABA and serotonin are the most most studied ones that are made by bacteria in the gut. And what we have learned, for example, with serotonin is that a large amount of the serotonin that is needed for healthy function that is used throughout, the body, especially in the brain, is actually made in the gut. And so essentially, the gut microbiome is one of our biggest partners here. Without the gut microbiome, without a healthy profile of these organisms in our guts, we're even affecting things like brain function, right? So this is, anyway, I geek out over that all the time. All right, so this is a very busy slide. I apologize. I will not try to make you understand all of it, right? But this provides basically just an overview of all of the ways that these previously discussed or listed metabolites I talked about basically regulate host metabolism. And the reason I wanted to bring this up is because regulation of host metabolism is one of the biggest underlying ways that affect our long-term health, right? So really understanding how the gut microbiome is doing this has been the focus of a lot of basic science researchers for decades now. And what we have learned is that there are multiple pathways. Of course, right? There's so many metabolites. So of course there are multiple pathways, but the gut microbiome affects our host metabolism. So I will just point out, for example, the TMAO that I mentioned earlier. TMAO is basically involved in the atherosclerosis pathogenesis, right? And so you can see that TMAO actually is related to it's produced by specific bacteria in the lumen of the gut. And I wish I could use a pointer. Oh yeah, there is a pointer. Perfect. So we have specific bacteria that are able to convert L-carnitine and phosphatidylcholine to TMAO, right? And this contributes directly to atherosclerosis pathogenesis, right? So that's one pathway, and we can see the myriad of them. There are so many of them that affect things like body weight gain and fat mass, and satiety has a couple of different pathways that I won't talk about today because this isn't an obesity or nutritional health webinar, but it's something I spent a bit of time studying. It's a very fascinating pathway worth studying. So what is in the gut? I've talked a lot about the bacteria, so that's no surprise that a lot of what we have in our gut is bacteria. And so when we look at the gastrointestinal tract from the stomach all the way to the colon, and we start to basically just count how much bacteria do we have in each of these, we start to see an increasing gradient as we go from the stomach to the colon. So stomach has like 10 to the 1 bacteria. By the time you get to the colon, you're at 10 to the 12. And the reason for this is because the stomach has pretty tough conditions for bugs to survive in. There's all of that acid. There's a ton of stuff going on in the stomach that bugs don't necessarily like. So there's some resistant organisms that are able to live there, but the large majority of them are in the colon where most of the fermentation of food happens. And so this is why, just as Scott mentioned earlier, when we take fecal material, poop, we end up measuring this 10 to the 12. It's largely because fermentation is occurring in the colon, and so they're happy there, they're able to grow quite a bit. All right, but it's not just bacteria. Fungi play a huge role in the microbiome, and this is beginning to emerge as one of the areas of the microbiome study. It's called microbiome on its own, because a lot of the papers, a lot of those thousands of papers referenced focus on the bacteria in the gut. But it really is a huge population of fungi as well. It's not that big or well characterized. And part of that is, technological limitations and method development needed and all of that. However, we know they're there, and they actually interact with the bacteria that live in the gut to perform some of the essential functions that are needed, and they're involved in multiple pathways. And so we can take something like Candida species, right? when we have an impaired microbiome, a lot of the time what we see is an increase in candida species. When we have a well-balanced, in quotes, and I use quotes, and I'll explain why later. When we have a well-balanced bacterial microbiome, we have fewer candida. And a lot of the time we've realized that increased candida is actually associated with diseases like type 2 diabetes and obesity. And a part of that is because of this downstream immune pathway that happens that we don't want, right? So it basically sets up inflammation, which isn't good for the body long term. So actually really important to note, the takeaway from this is that indirect interactions between gut fungi and bacteria, they actually affect our host immunity by changing the cytokine profile to a more inflammatory state, which can contribute to diseases. All right, so what affects the composition of the gut microbiota? Diet and lifestyle are the biggest drivers. This is why it's bolded up here. And Scott mentioned diet quite a bit, talked about how that, you know, they base the selection of the donors on diets. And so they have the omnivore and a vegetarian, and that's how they collected the stool for the standard. This is because, again, diet is probably one of the largest drivers of the gut microbiome. And you can see why, right? We have so many different things in food that can inform what is in the gut. But there are also other factors. There's host-specific factors, such as genetic factors. There's age, there's the overall health status of the host. And so, you know, different diseases can affect the microbiome. And then there's consumption of drugs, including antibiotics. So these are the largest drivers for the gut microbiome. Here, I'm just showing how diet affects the gut microbiome over time. The largest takeaway from this slide is that as we go from being a baby to an adult, where the diversity of our food is changing, right? So when you're a baby, you're only taking like breast milk or formula or both. You're really limited in the amount of food or different types of food you're taking. You have a very small, low diversity of microbiota down here. And so you can see that the different colors, there's fewer. Then in the background here, as diet diversity goes up, and the diet diversity is the upper panel, as that goes up, you begin to see that there's an increase in the diversity of different microorganisms that are inhabiting your gut. So these types of studies are some of the biggest. They basically give us the most information around how diet affects the microbiome, helps us to see that really changing the diversity of your diet is what is impacting your microbiome long term. So here is also a really cool summary that I found from a paper. And here it's just showing that if you take a diet high in animal fat on the right, so a diet high in animal fat and protein on the right, but low in plant fiber. So this is what we call a typical Western diet in those of us who study type 2 diabetes and obesity. And you compare that to people, or basically a gut microbiome of an individual that takes a diet low in animal fat and protein, but high in plant fibers. We see things like the metabolic process of each of them is different. For example, the person taking the Western diet, which is high in fat, is going to have an aberrant microbiota, which we find typically associated with metabolic diseases like type 2 diabetes and obesity. And this is typically present even when the person doesn't have any of those conditions. It's largely because of the diets that they're eating. And then you begin to see the downstream effects of that. We see the impact on the lumen, for example. We see impaired barrier function, which is commonly referred to as leaky gut, allowing certain things in the gut to get out and set up systemic inflammation, which just low-grade systemic inflammation by itself can go back into the cycle and end up creating downstream effects later on in life. And then antibiotics, I mean, this is probably predictable to a lot of the people watching this, right? It's antibiotics. They're set up to kill bacteria, right? So what ends up happening though with antibiotic consumption is that it affects the overall stability or homeostasis of the gut microbiome. And so say you have one infection, you know exactly what bug you're trying to eradicate and you take antibiotics. You aren't just eradicating that one bug. There's what we call collateral damage. And so here we start antibiotic treatments. This is a line to show, and these blue dots basically show the number of bacteria present in that individual. So before antibiotic consumption, you can see that's pretty high number of bacteria. And then they take antibiotics, you can see that rapid drop in the bacterial number. And this is after antibiotic treatment stops, we still have a low number of bacteria. It takes a really long time for that number to recover. So the take-home message here is that it takes a while to recover from antibiotic treatment, and so one of the largest things we need to be aware of is when we are prescribing antibiotics as clinicians or we're consuming them, just engaging in that conversation with patients or with our healthcare practitioners to try to mitigate some of that damage after taking the antibiotics, because we need antibiotics to survive. We can't live without them. But we can try to mitigate some of that damage by making sure we're prescribing more specific antibiotics to treat exactly what we have. And number two, having a discussion with our medical practitioners to work out a plan for making sure we restore some of this balance early. All right, so now I'm gonna basically just start talking about how we get a microbiome in the first place. And I'm gonna try to speed through this part pretty fast. We know that age affects the microbiome structure. The way we know this is by doing longitudinal studies. So we take people and study them for years, or we take samples from a homogeneous population of people where there are different age groups represented. And so this is from a review paper that summarized some of that. What we see is that there is a change in the relative abundance of different types of bacteria in the gut of individuals based on their age. And so when we have a preterm infants at birth and discharge, they have, for example, fairly low amounts of bacteroidetes. And then over time, you can see as they grow, that's the yellow line, they're having more bacteroidetes. So we know age affects the microbiome, but how do we get a microbiome in the first place? Where does it actually come from? We actually get our microbiomes from our mothers. So during pregnancy, there is a hypothesis that the womb is not sterile. So some bacterial interactions may be happening with amniotic fluids. This is still really early stage, so this is not represented on the slide. But starting right at birth, depending on the mode of delivery, we start to see different types of bacteria that are colonizing the guts of children. So this gives us a really good indication then that we get most of our bacteria from our moms. I will just show this real quick. The part to focus on is this lower graph right here to, again, just confirm that we get most of our bugs from our moms. When we look at a newborn, when you're born and you basically have very low bacterial diversity, as time goes on and by 1 year old, your microbiome is expected to resemble a more mature microbiome. By 1 year old, most babies' microbiomes will begin to look almost exactly or identical to their mothers. And this is largely dictated by a whole bunch of different things, including breast milk. So if a child is breastfed, for example, there is some evidence that bacteria can be transferred from breast milk to babies. There's also the delivery route. We already talked about that. So for example, babies delivered through C-sections have bacteria that are more representative of the skin than, babies that are born vaginally. Those have bacteria representative of the gut. So basically based on mode of delivery. And then a lot of the time we're beginning to learn that the health status of the mom can affect the baby's microbiome as well. We'll talk a little bit about that later. So many factors can really affect the colonization of a baby from birth. There are prenatal factors, there's neonatal factors, and then there's postnatal factors. So after a child is born, most of the time, the postnatal factors are the ones that come into play. And as we might imagine, diet becomes important. And so for a baby, that's breast milk versus formula, the geographical location, the family members, the interactions of a baby with a community. So is that baby being passed around, or is the baby mostly isolated and with just their mom and their dad? Maternal diets plays a huge role because, you know, again, we're passing some of that from the mom to the baby, and then the weaning, what they eat actually after you wean them off. So this shows a window of opportunity for microbiota modulation in children. For those interested in pediatric modulation, typically as the child gets older, we lose that window of opportunity. So we want to do it earlier, probably right after birth. All right. And here is a very busy chart again. You don't have to look at all the details. This is just to point out that the delivery mode affects the transfer of these microbes from mom to baby. So these are vaginally born babies up here. These are vaginally C-section delivered babies down here. And the big take home here is that the vaginally born babies typically have similar species of organisms, the same levels of bacterial species as their mothers than the C-section babies. So C-section babies tend to have different levels of the same organisms, and each of these represent a time point. So the C-section babies, it takes them about a year to get to the mom's level. For vaginally delivered babies, a lot of them resemble their moms right off the bat. Okay, so nutrition. Undernutrition was a big focus of study for a while. And the reason for this is because undernutrition plays a role in the formation of the microbiome. And so there are multiple studies that have demonstrated that the first 1,000 days of life is the most critical period for making sure that we establish a healthy gut microbiome. And actually, it helps to determine the health status of the individual later in life. So what we're showing here on this graph is just some of the key bacterial taxa that affect the child at different time points, that play, you know, roles at different time points and different developmental milestones and the first 1,000 days of life. And so just going all the way to like weaning up to day 450, this is the list of organisms that we hypothesize are important to have at this stage because they affect nutrition and hygiene. And then, so you can then also see the organisms that are associated with poor nutrition, undernutrition, and poor hygiene and things like that. Okay, so there is a hypothesis called the developmental origins of health and disease. This basically posits that there are critical periods in fetal and early development in a child's environment which affects a child's metabolism, neurogenesis, and basically just long-term health outcomes. And we're beginning to realize in the microbiome field that, you know, the risk of chronic disease can actually be associated with the type of microbiome that babies start out with, right? So there's a lot of companies who are beginning to study this a lot, trying to figure out if there's a way to modulate the microbiome of babies right off the bat, right here, when there is a reduced risk of chronic disease before later in life, they start to have that higher risk here. Okay, so here are the important factors by critical periods in a child's life. During pregnancy, the maternal health status is probably the largest contributor, along with the vaginal health of the mom. And then after pregnancy, you know, delivery, the mode of the delivery and the gestational period, along with hospital environment, play the largest roles. Then during infancy, that's when actually host genetics kicks in. So it's pretty cool to see that most of the things before infancy are environmental and based on the maternal health status. So many things determine long-term health. We've got genetics, diet, mode of delivery, we just talked about that, environment, drugs, exercise. There's so many things that impact health. So we're just gonna talk briefly about some of them. So the gut microbiome early in life, I just mentioned, can predispose individuals to illness later on. So this is a summary from a review article that demonstrates that there are dysbiotic communities, or when we use the term dysbiosis, we're referring to an unbalanced community of microorganisms present in babies that can determine if they're going to get certain diseases later on. And this has been linked to things like neurological disorders, allergies, cardiovascular diseases. Cardiovascular disease is probably the largest studied areas out of these. I'm going to skip that because I don't want to spend too much time on it. So what are the maternal factors that actually affect a baby early in life that can determine their long-term health aspects? One of the largest is the mother's weight. The second is diet. Third, consumption of the drug intake, and then the environment they live in. So there are things that are not within mother's controls, and then there are things that can potentially be mitigated. But a lot of the time, these things are not within the baby's control, right? Because it can affect the baby from the mother, so they can't control any of these. But what we know is that all of these can lead to a dysbiotic gut microbiome in the mom, which can then get transferred to the child. And this can actually affect fetal brain structure and function. And so we can start to see things like, again, increased metabolic stress, increased hyperlipidemia, increased fatty acids, all of those things. These can actually affect the development, the healthy development of the baby. All right, so when we have I mentioned weight plays a role here, the weight of the mom. The overweight or obese mothers have a dysbiotic gut, and a lot of the time, not all of them obviously, but most of them do. And what we have found is that there is leaky gut, which I talked about earlier, just impaired barrier function. This can actually divert the transmission of bacteria that are obesogenic to a baby, right? And this can then lead to an altered gut microbiome and immunity, which unfortunately can lead to a higher risk of childhood overweight and obesity. And the mechanism by which this happens is typically through LPS and then production of an impaired profile of short-chain fatty acids. All right, this has also been seen in NAFLD, which is non-alcoholic fatty liver disease. We see that when we have maternal obesity being one of the factors, along with mode of delivery, antibiotic exposure, or breast milk exposure, we can actually build a model that can predict for the development of NAFLD or NASH in a child later in life or not. And a lot of it is largely, again, due to increased gut permeability, which leads to inflammation downstream. All right. Okay, so I've talked about impaired barrier function, but I just wanted to show a brief graphic of this. When we say impaired barrier function, what we are referring to is that these epithelial cells in the gut actually have really tight junctions. They typically don't allow just anything to get in and out of the gut. And so if this is inside the gut and this is outside the gut, stuff that stays here, it's only a selected few can make it through this barrier and get in a healthy individual or a non-dysbiotic individual. But we do have conditions where, because of an impaired gut microbiome, a microbiome that is dysbiotic or unbalanced, we then begin to see this barrier, unfortunately, become less tight, which then allows stuff to leak out of the gut. This is what we call leaky gut, and then cause systemic inflammation. All right. So how can we benefit from all of this information I just shared? The largest takeaways for me are that we need to start having conversations around how we can improve the long-term health outcomes of individuals, starting from when they're children or when they're babies, right? Even starting with maternal health, really, right? Because if we can then, if we can ensure or improve the healthcare outcomes of mothers, we know based on evidence that this can actually impact the healthcare outcomes of children. And a lot of the things that can impact the healthcare of mothers then leads us into the conversation around healthcare disparities. And a lot of it is stuff that is not within the control of the mother. So there's macroeconomic conditions, there's public health and health policies, there's culture and values, and a lot of things that determine health in general that are not within the control of individuals. Even gender, sometimes income and education are things that are out of reach for individuals. So, again, from a policy perspective, being able to control some of these things can actually help improve healthcare equity. Okay, so we know in microbiome that differences in composition can result from ethnic individuality. And a lot of these are factors to do with diet, lifestyle, socioeconomic conditions, cultural practices, and genetic ancestry. And this information is largely from large cohort studies from across the globe. So notably, we know that there's also increased risk for certain conditions based on your ethnicity. And so if you put these two together, you can begin to see the potential that the microbiome has for being able to provide personalized medicine approach or personalized healthcare interventions for specific ethnic groups based on the microbiome profile, based on their diets, their lifestyle, based on socioeconomic conditions. You can begin to craft a really specific way of caring for different groups of people, which can help to then reduce the gap, right, for healthcare equity. And so just as I mentioned, we can use ethnicity-associated microbiome composition to link the influence of factors like ethnicity-associated dietary or cultural patterns with healthcare disparity etiology. And this can help with policy. This can help even in the hospital, in clinical practice. So again, while individuals and governments are working right now to address the public policy, societal factors that underlie health disparities, we have a role. If you're a researcher in the audience, we have a role to play to leverage some of the studies we perform by prioritizing microbiomes by ethnicity and disease by microbiome associations to build a foundation for therapies that are for specific ethnicities. And then this is a quote from Andrew Brooks. It was an editorial that I found particularly compelling. And what he said is, you know, as a culture, addressing injustices in access to healthcare and fresh food, socioeconomic mobility, and many other factors underlying disparities is an ideal, right? It's ideal, but it's long-term, it's protracted to pursue equality in health outcomes. So right now, what can we do? Practical approaches that leverage these ethnicity-associated microbiomes for clues about contributing factors to increased risk for disease can help to provide, you know, lifestyle interventions for health disparities that are worth pursuing. So that you know, is pretty powerful to me, being a microbiome scientist, to know that we can actually utilize some of this in that way. Okay, so how can we benefit from all of this just across the board? Scott talked about this, microbiome-directed interventions. We can have untargeted interventions, and Scott talked about probiotics, right down there, and then probiotic foods. There's also prebiotics, which typically are foods or supplements. There's postbiotics, there's symbiotics, there's a lot of biotics, as you've heard, but typically there's also fecal microbiota transplantation, which we're using currently to treat Clostridioides difficile infection in a chronic way. So this was just approved by the FDA a few months ago as the first therapeutic. And then we have individualized nutrition. So we can actually take information based on your microbiome and build a personalized nutrition profile, which would be, you know, easier to hopefully adapt to because it's customized to you. Exercise is also one of the ways we've shown we can actually target the microbiome. And this leads to general improvement in microbial composition and functions. But there's also targeted approaches, genetic modification of strains to perform certain functions, such as work as an antibiotic. So instead of taking an antibiotic chemical, you would take a bug or a bacterium which has a phage to eliminate an infectious disease. That's one of the pathways, but there are multiple targeted ways to treat some different disease indications. So in conclusion, I would like to quote the father of modern medicine, Hippocrates, and say that, you know, just as he said, all disease begins in the gut, and he said this so long ago, it's true today. We can say that a lot of diseases begin in the gut, and our food should be our medicine should be our food. Why? Because, hey, the diet is the largest determinant of your microbiome. So you can control that piece, at least you can make a difference. All right, thank you. No questions now. The questions can come